This is May 16th, 2020. These are some audios to go along with the 3 PowerPoints that just hit a few of our final exam, our lecture final exam topics. So earlier we had started on the second PowerPoint. We did slides 1 through 27, which was Unit 4 Metabolism. And now we are on slide 28. This is the start of the microbial growth, which will be relatively short. And we can see here, you guys, I think we only have like maybe six slides for microbial growth. And then it'll run right into microbial genetics part 1. Okay, so hopefully we'll get this all in one recording here. Okay, so let me see if I can't go to full screen. Okay, so you guys, again, this is on microbial growth, and our focus was primarily on bacteria. So bacteria reproduce asexually, meaning there's a single parent. They're going to copy their chromosomal DNA, and then they're going to split in two in a process called binary fission. So one cell divides into 2 cells, and the time it takes for one cell to divide into 2 cells is called the doubling time or the generation time. And bacteria are hardwired for this, meaning it's, for example, E. coli under ideal circumstances, E. coli can divide every 20 minutes, so a generation time of 20 minutes. But in contrast, say Mycobacterium tuberculosis, it might be hours, maybe even days, right? And that's just their biology. Yeah. Because of E. coli's generation time, short generation time, they've been kind of the ideal organism to study, say, metabolism and genetics in the lab. So when we are studying microbial growth, say bacterial growth, we're not looking at the gain in weight of a single bacterium or an increase in size. We're usually looking at population sizes. So these would be lab experiments. So we'd start out with sterile broth, say in a flask, and we would introduce a few bacteria. Let's just pretend maybe it's E. coli, and then we would incubate, and then time intervals we would sample the broth, and we would calculate the number of living cells, viable cells, per mil of broth, and then we would plot those data points and then use the line of best fit to get our graph here. And you recall, folks, that like with E. coli, right? Once it starts dividing, it doubles every 20 minutes, and the population sizes can get really high, like 10 to the 8th, 10 to the 9th bacteria per milliliter of broth. We said if you use regular arithmetic paper, graph paper, it wouldn't work. So we use semi-log paper, in which the x-axis time is regular arithmetic scale and the y-axis is a log scale. So this would represent the log base 10 of the number of living cells or viable cells per mil. And in using the semi-log paper, then we end up with this standard bacterial or standard microbial growth curve, and there would be 4 phases. And again, you guys, this would be like a lab experiment where we don't introduce new media, we don't remove any of the spent broth. So it's a closed system, right? So we start out with this lag phase where there's no apparent increase in the number of cells, but during this time the cells are adapting to their new environment. They're transporting nutrients into the cell, they're synthesizing the organic molecules they need, they're copying their DNA, and then suddenly all the cells start dividing at once, right? And this is called the log or exponential phase. So this is when we would calculate the generation time or doubling time. It's the time when the cells are most sensitive to antimicrobials or antibiotics, right, in the log or exponential phase. It's the best time to do a gram stain because a gram-positive bacteria, their peptidoglycan shouldn't have any holes in it yet. As we said, it's the best time to calculate the generation time. And then in this closed system, right, we're not adding fresh broth. We're not getting rid of any of the kind of broth that has a lot of metabolic waste products in it. We would start to see the number of cells dividing start to slow down a little bit. And indeed, during this next phase, the third phase, stationary phase, the number of new cells arising from binary fission is going to equal the number of cells that are dying, right? Because even bacteria have a lifespan. So During stationary phase, then, one could say this represents the carrying capacity of the environment, right? And other really important things you guys want to remember, that very often bacterial pathogens living in biofilms in your patients, they're in the stationary phase of growth. And they're naturally more resistant to antibiotics in the stationary phase of growth, in addition to the layers of the biofilm acting as a kind of protective barriers against antibiotics and phagocytes and antibodies, right? And furthermore, folks, if we were doing this experiment on an endospore producer, say Bacillus anthracis or Clostridium botulinum or Clostridium tetani, we would probably start to see the production of endospores probably in late log. Because you'll remember, you guys, endospores, their function is to protect a copy of the chromosomal DNA when environmental conditions get bad. And here towards late log, nutrients are going to start to decrease. We're going to have an accumulation of toxic metabolic waste products. So definitely the environment is starting to deteriorate here. So we'd probably see the onset of what's called sporulation, producing the endospores in late log. And then endospore production would continue through stationary phase. And of course, we'd see the greatest number of endospores during the fourth phase, the death or decline phase. And in the death and decline phase, the number of cells dying outnumbers the number of new cells arising from binary fission. And we would predict, right, if we just left that culture in the broth, say if it was E. coli, maybe in a 2 or 3 weeks, all E. coli would be dead, right? So let's take a look down here, some of the notes. So we want to remember that if you're looking at, if you're working with gram-positive bacteria, you would have, you would start to get false gram-negative reactions in the stationary phase and the death or decline phase, because once the bacteria hit stationary phase, there are enzymes that continue to cut the peptidoglycan, right? But the bacteria they're no longer growing, and they don't repair the holes in the peptidoglycan. So gram-positive bacteria, as they age, their peptidoglycan literally becomes holy. They can't hold on to the crystal violet iodine complexes. They'll decolorize when you add your alcohol acetone, and then they'll counterstain with your safranin as pink. So we get false gram-negatives and stationary in the death phase. And as we said, you guys, also in the stationary death phases, we would predict we'd see the maximum number of endospores if we were working with a bacillus or Clostridium. And then another thing when we're talking about microbial growth is we need to know what do they require? Is their energy source? Excuse me. And what is their carbon source, right? So we can classify organisms based on these two characteristics, energy and carbon source. So we said for our exam, you guys, you don't have to know all 4 classes. We're just going to focus on the photoautotrophs and the chemoheterotrophs. So photo, this first word is telling us what is the energy source. So photo, we know light is the energy source. Autotroph is telling us what's the carbon source. So autotroph literally means cell feeder. And this is because photoautotrophs can use inorganic carbon dioxide in the air as a source of carbon to build their own organic molecules. So photoautotrophs would include our little guys that make chlorophyll A, carry out oxygenic photosynthesis, plants, algae, and cyanobacteria, right? But in addition, folks, now that we know about anoxygenic photosynthesis, we have to include the anoxygenic photosynthesizing bacteria, like those cool purple sulfur bacteria, and there's these cool green sulfur bacteria. So they don't make oxygen, but still light is their energy source, and they use carbon dioxide as their source of carbon to build their organic molecules. Then the other group, folks, that we wanted to remember, this is the group to which we belong. And again, any cellular microbe that invades another organism and causes harm, they would also all belong to this group, the chemoheterotrophs. And chemoheterotrophs require preformed organic molecule both as an energy source and as a carbon source, right? So for example, folks, that's why we have to eat food, right? Our food are those preformed organic molecules. So we said that the chemoheterotrophs, again, they have to have preformed organic molecules. And we had discussed 3 different ways they can use those organic molecules as a source of energy to drive ATP synthesis. So we talked about aerobic respiration. Most animals, fungi, protozoa, and many bacteria, anaerobic respiration. Some animals, I have to come back here, you guys, because I'd like to check this out. Some animals, protozoa, and bacteria, and fermentation, some bacteria and yeast. And we could argue, you guys, our cells even know how to, for a very short period of time, can switch to lactic acid fermentation under anaerobic conditions. If we're in the lab, we also need to know if a microbe is fastidious. And fastidious microbes are those that either have really simple enzyme systems or defective enzyme systems because they can't make many of their organic building blocks. So consequently, we have to, if we're trying to grow them in the lab, we have to give them really rich media, enrichment media like blood agar. It's really important that we include growth factors like amino acids, vitamins, precursors of nitrogenous bases, right? And the question might be, well, why are so many pathogens that invade another organism and cause harm? Why are they fastidious? Well, again, folks, it just goes back to they need a lot of preformed organic molecules, right? Because they can't make them themselves. So again, if you're working in the microbiology lab and you receive a clinical sample from a patient and you suspect that it might be a fastidious bacterial pathogen, you want to make sure that you culture the sample on really rich medium like blood agar. And then, folks, we said that in discussing microbial growth, that microbes have a specific relationship to oxygen, depending on whether they have evolved protective enzymes to protect them from those toxic reactive oxygen intermediates such as superoxide anion or radical and hydrogen peroxide, right? So organisms that are what we call obligate aerobic organisms, like we are obligate aerobic organisms, right? We have to use aerobic respiration to make enough ATP to survive long term, right? We said we can, for a short period, switch over to lactic acid fermentation, but can't survive very long. So organisms that have to use aerobic respiration to make ATP, we'd call them obligate or strict aerobes, right? So we know they're going to be using oxidative phosphorylation and aerobic respiration. They can't carry out fermentation or anaerobic respiration, right? So you recall that we described this experiment, and then if we had been in lab, we would have run this experiment. This would be thioglycolate medium, and thioglycolate reduces molecular oxygen. So you would have a little bit of oxygen up here at the top, but as you go down to the bottom, this would be totally anaerobic. So we would inoculate our different bacteria and incubate them, and then look at the pattern of growth. So stricter obligate aerobes like Pseudomonas, they're only going to grow up the top here, right, where there's enough oxygen to carry out aerobic respiration. But remember, you guys, when the first cells evolved here on Earth, the atmosphere was anaerobic, right? And so when the primitive cyanobacteria started making oxygen using oxygenic photosynthesis, oxygen can actually be really toxic to organisms, right? The oxygen can interact with enzyme systems, or like when electron transport chain evolves and we get those toxic reactive oxygen intermediates that can kill. So if you are going to live in an aerobic environment, and especially if you're going to use oxygen in aerobic respiration, you had to have evolved protective enzymes, right, to detoxify those reactive oxygen intermediates. So we said obligate or strict aerobes like ourselves, we're going to be making superoxide dismutase to detoxify the superoxide radicals or anions. Also is a product of the superoxide dismutase, you're going to make hydrogen peroxide. So usually stricter obligate aerobes will have another enzyme. For example, we humans have catalase to break down the hydrogen peroxide into water and molecular oxygen, right? So again, you guys, if we're going to be an obligate or strict aerobe, we have to have protective enzymes to break down those toxic reactive oxygen intermediates. Now, if we go over here and we look at the genus Clostridium and we ran this experiment, we would see they have the opposite growth pattern. Clostridium can only grow in anaerobic environments, so we call them obligate or strict anaerobes. And the reason is they never evolve those protective enzymes, right? So they can't live in an aerobic environment because they lack the superoxide dismutase, they lack the catalase and or peroxidase, yeah? Okay. And with Clostridium, you guys, it's like in the medical micro section, we talked about, how the endospores of Clostridium, they're everywhere in the dirt and the soil. So you want to remember that the endospores of Clostridium are not destroyed in aerobic environments, and that's how Clostridium can survive, say, on that rusty nail, in the soil that you step on and then end up with tetanus. Okay. And then folks, over here is E. coli. And you might remember from the earlier video that we said that E. coli is so metabolically adaptable. If there's no O2 present, it can carry out fermentation, or if there's an alternate terminal electron acceptor, it can switch to anaerobic respiration. And then if oxygen is present, it can use aerobic respiration. So since it uses aerobic respiration, then we're going to presume that it's going to make the protective enzyme superoxide dismutase, and in this case catalase, and we would be right. Because microbes make the most ATP using aerobic respiration, we would see this uneven distribution of cells with what E. coli, which we call a facultative anaerobe. It can grow anaerobically or aerobically. With aerobic respiration, they're going to make maximal ATP, so we'd have higher concentrations of cells in the aerobic zone. And then when they switch to anaerobic metabolism, they're going to have lower ATP production, so we're going to see lower concentration of cells in the anaerobic portion. Then, folks, this is really cool. Members of the genus Streptococcus, which are part of that big family, the lactic acid bacteria family, that can only carry out lactic acid fermentation, right? They never, my understanding is they never evolved cytochromes, so they can't carry out any kind of respiration. They don't have an electron transport chain. So all they can do is carry out lactic acid fermentation, right, with only a gain of 2 ATP per glucose. So with them, we would see a even distribution of cells throughout the broth, aerobic or anaerobic. All they're doing is carrying out lactic acid fermentation. And the key is they do have at least one protective enzyme. So in the genus Streptococcus, they don't make catalase, but they make another enzyme called peroxidase that will help detoxify hydrogen peroxide. So these little guys are called aerotolerant. They can tolerate air anaerobes. They can't use it. They can't use the oxygen, right? But they can tolerate living in aerobic environments. And then the last one, and I don't think we have a slide for this one, the microaerophiles, the Campylobacter, they grow at oxygen concentrations lower than atmospheric levels. So I think the atmospheric oxygen is like around 21%. So Campylobacter will grow at a little bit lower. I'll just make this up, you guys. Let's say at 15% oxygen concentrations. And we said that was fascinating because Campylobacter, they're a real common cause of foodborne infections. They're fecal pathogens. So let's say if we're working with a raw chicken or a raw turkey, there's a good chance the outside of the carcass and inside the cavity is also contaminated with fecal pathogens like Campylobacter. And there was one German research group who showed that it's through making a biofilm that Campylobacter can adjust the oxygen concentration when they're growing on the surface of raw poultry. And that's how they create this little perfect environment where it's just the right amount of molecular oxygen. I thought that was pretty cool. So let's see here, you guys. Why are some microbes obligate or strict anaerobes? Remember, it's because they can't make any protective enzymes, no superoxide dismutase, nor catalase, nor peroxidase. Which enzymes do aerobes produce? So the aerobes are the obligate or strict aerobes are going to make those protective enzymes. And we said that Clostridium can survive in aerobic environments by making the endospores. And then furthermore, you guys, in addition to classifying microbes based on carbon and energy source in relationship to oxygen, we can categorize them according to their optimal, the optimal temperature for the highest growth rates. And we said you guys did not have to memorize these temperature ranges, but we did talk about 4 different groups. The psychrophiles, literally cold lovers. So these are guys that will like grow very well in your refrigerator. Most pathogens, cellular pathogens that are going to invade humans are going to be what we call mesophiles. So for moderate temperature, and again, they're going to grow best at around human core body temperature, about 37 degrees Celsius. And then there's microbes that have evolved in hotter environments, so we call them thermophiles. And these are the wild ones, you guys. And then there's some microbes that have evolved in really, really hot environments, like near active volcanoes or hot geysers. And you guys, 100 degrees here, that's boiling. So you can see there's some microbes that can live, in conditions that would definitely kill us, boiling, right? The hyperthermophiles. And there are some bacteria that are hypothermophiles. Some of the archaea, and you remember the archaea sometimes are called extremophiles. Some of the archaea would be considered hypothermophiles living at these really high temperatures. With the psychrophiles, you guys do remember Listeria Monocytogenes. So it's a gram-positive bacterium. It can live in the intestinal tract of a variety of animals, including cows and goats, I know. So one of the challenges is if pregnant women drink milk that has the Listeria monocytogenes, and it can also be introduced in milk, you guys, through white blood cells. So milk has white blood cells in it. So Listeria monocytogene might be, for example, growing within the macrophages found in the milk. So either through fecal contamination or through the white blood cells in um, the milk can have listeria in it. So if a mom's pregnant and she drinks like a raw milk product or eats a raw milk product like a cheese or any milk that hasn't been pasteurized, a Listeria monocytogenes may be present. And so mom gets infected and the listeria can cross the placenta, invade the baby, and can cause a spontaneous miscarriage or can cause neonatal sepsis or meningitis in the baby. So I really, really recommend that pregnant women make sure that you're consuming pasteurized milk products. And then furthermore, folks, what makes this wild, like let's say there was an outbreak of Listeria monocytogenes in the community, and so you're taking milk samples or dairy products and you want to try to grow the listeria, what you can do is put the milk sample, the dairy product, in the fridge. And that the low temperature should inhibit the growth of most other microbes. But remember, listeria can switch from being a mesophile in humans, it can switch to being a psychrophile. So you can actually enrich for the presence of listeria by putting your milk or dairy sample in the fridge. And that's called a cold enrichment, which I just think is wild. And then, folks, we had also talked about classification of microbes based on the pH range in which they like to grow. So we talked about acidophiles, acid-loving bacteria, neutrophiles. So most human pathogens that invade us are probably going to be neutrophiles, right? They grow best around pH 7. And then there's some alkalinophiles, Vibrio Cholera. Fascinating natural history of Vibrio Cholera, but it's really flexible, right? So obviously it can live within humans. It causes cholera, right? But then in its natural reservoirs, it can live in waters that are like an alkaline pH, which is again pretty wild. And then, folks, we had talked about osmotic pressure. You know, we had always, way back in cells, we had talked about hypoosmotic environments and how microbes that don't have some strategy to deal with the influx of water can die from um, osmotic lysis. But then, historically, our ancestors, and even today, we can purposely create hyperosmotic environments by using sugar or salt. And in a hyperosmotic environment, microbes will dehydrate, right? There'll be a net movement of water out of the cells. And without the water, we want to remember, metabolism usually doesn't proceed very well. So by salting or adding sugar to foods, this is a way to preserve the foods in the absence of refrigeration, right? You're going to inhibit the growth of spoilage microbes. Isoosmotic is when the solute concentration inside and outside the cell are the same, or another way of thinking it, the water concentration inside and outside the cell are the same, right? And then we did talk about some of the archaea are what we call halophiles. They're salt lovers, right? They've tried to return these to a natural state, but down in the Bay Area, they used to have like these salt ponds, right, where they let the water evaporate from the seawater to harvest the salt, and they would be these beautiful, gorgeous colors, like if you were flying over them, maybe coming into SFO, beautiful, brilliant colors, and it was from the archaea, these halophiles that had these gorgeous colors. And we did say, you guys, that Staphylococcus aureus, that can make this powerful enterotoxin, they can live at really high salt concentrations. You remember the mannitol salt lager, 7.5% weight per volume, 7.5 grams of sodium chloride per 100 mils. Staph aureus, since it's evolved to grow on our skin, which is salty, right? Staph aureus can grow in really high salt concentrations. So he said, for example, like if you use salt maybe to preserve maybe a meat, you got to be careful because Staph aureus could still grow there, producing the enterotoxin. which is a powerful exotoxin. So if you consume the meat that has the Staph aureus enterotoxin, it causes that projectile vomiting that's so dramatic. And then, folks, that leads us into microbial genetics. And again, I think what I'll do, just trying to chop these videos up, I'll stop here and try to get this up to YouTube and post it, and then I'll return and do the microbial genetics audio. And folks, hopefully I can get to the third PowerPoint today, and if not, today's Sunday, tomorrow's Monday. Our first final exam is Tuesday, so I apologize I didn't get these audios made sooner. But if not tonight, I'll do the audios for the final exam items, the third PowerPoint. I'll try to get that done tomorrow morning. I do have some makeup exams going on, and I like to be on the computer, so if folks are having any problems, but I'll try to get the audio for that third PowerPoint posted as soon as possible tomorrow. Okay, thanks for your patience, you guys, and let me close this one down, and then I'll come back and we'll do another audio for the microbial genetics part 1. Exam item.